Hello. So we are talking in this opportunity with Dr. Patrick Pearson, who is a postdoctoral research associate at the University of Massachusetts Amherst. He earned his Bachelor of Science degree at the University of Vermont in 2010 and his PhD in microbiology at the University of Massachusetts Amherst in 2023. His PhD work focused on Borrelia burgdorferi, which is a causative agent of Lyme disease. As part of his PhD research, he developed an assay that can detect the many genotypes of B. burgdorferi. In general, Patrick is interested in research on ticks and tick-borne diseases. Today, we will be discussing his lab protocol article published in PLOS ONE, A Borrelia burgdorferi outer surface protein C genotyping method using Luminex technology and the associated protocol posted in protocols.io. Patrick, thank you so much for being with us.
Thank you.
So can you briefly explain what this protocol is about, how to use it, and why it is relevant?
Yeah. So as you mentioned, Borrelia burgdorferi is a tick-borne pathogen and the causative agent of Lyme disease here in the United States. And that's what this protocol focuses on. It focuses on Borrelia burgdorferi. So just a little bit of background. The way Borrelia survives, or the way it exists in nature, is it infects different reservoir hosts, things like mice and chipmunks, at least here in the Northeast. And then it's transmitted from host to host by a tick vector, so Ixodes scapularis in the Northeast or Ixodes pacificus in the West Coast. But Borrelia burgdorferi isn't homogeneous, so it's made up of many different types of variants or genotypes. And one way you can tell these apart is based on differences in the sequence of one very polymorphic gene, which is called outer surface protein C, or the OspC gene. You can use those differences to define different variants or different strains. And so the point of this protocol is just to detect those different OspC genotypes. And so we use technology from this company called Luminex, which is based out of Austin, Texas. We use their reagents, we use their instrument, and then we tweaked it for our purposes. And it's composed of essentially 5 parts. You do a PCR reaction, you clean up the PCR reaction with exosat, and then there's a step called allele-specific primer extension. And basically what that means is any alleles that are present in that PCR reaction are then specific primer binds. There's an extension step, and biotin-labeled nucleotides are incorporated. So you're just labeling any PCR products there, any different OspC genotypes. You couple those to a microscopic magnetic bead. You add a reporter solution, and then you measure fluorescence on the Luminex analyzer. And so it's relevant because what you can do with this assay is you can ask and hopefully answer very basic biology questions about Borrelia burgdorferi, like what variants are in ticks and hosts, and also what variants might be infecting people. So it has some public health significance as well. So what I like about this assay is that a lot of Borrelia research focuses on presence-absence, if it's there or not. This assay just has a finer resolution where we can say it's there, and then we can also tell you what specific types of Borrelia are present.
So can you then describe what was the state of the art when you decided to develop and optimize this protocol? Was there anything already published that would allow you this refined assessment of the different genotypes of Borrelia?
Yeah, so sort of like so there's a few different assays, but really the industry standard, I think, would be this one assay called reverse line blot, or RLB, for OspC typing. And we actually drew a lot of inspiration from that assay. So we used a lot of the same primers and a lot of the same, the same primers from that assay. We sort of co-opted them for this assay. And so there is this assay called reverse line blot. It's a DNA hybridization assay that has worked for us in the past, meaning we as in the lab. So people in the lab have used this assay. successfully. We've gotten good results. We've published data and papers using that assay. But what we thought we could do is potentially make a better assay using this Luminex technology, and better in a sense, at least in two aspects. One is the RLB assay isn't as high throughput, so I think you can only do about 40 samples at a time. Our assay, we designed it to be 96 well plate compatible, so you can do 96 well plates and you do multi-channel pipetters, so it's a little bit higher throughput. And also, full disclosure, I've actually never done the RLB assay, but one thing I know about it is the readout or the output of it is an X-ray film of the blot. And what you do is you look at it and you kind of look for the blots or the dots, and you go, okay, it's present or it's absent. But sometimes the dots can be like very faint or small, and you're sort of squinting and you're going, is it there or not? And so one thing I like about our assay is it gives you a numerical value. So you can go, if it's above this value, you can draw a cutoff. You can go, if it's above this value, it's positive, and if it's below this value, it's negative. So I think it's less ambiguous. So there are other assays, but we thought we could design something a little better. And I also have to point out that it wasn't just me; it was other people in the lab had their hands in this, and I sort of came into it and tweaked it and sunk my teeth into it. It was one of the first projects that I worked on. But really, we thought we could make a little better assay, and then we could add it to our toolbox. As one of the tools in our toolbox for testing those samples, primarily tick samples. And then we could test hundreds and thousands of tick samples and have really high confidence in the results.
That was a great explanation. But after all the time you invested in developing this assay, what was the motivation for you to create a nice format to put on the platform of protocols.io and then go one step further and have it peer-reviewed as a formal index paper?
Yeah. So primarily just for transparency, right? So have a really detailed protocol and then have it very transparent on this protocols.io platform. And one thing I like about the protocols.io platform is you can put in like the catalog numbers of everything. So all the reagents that you buy, you can put in those catalog numbers, because that's something that I've noticed with other papers is they might say they use a certain reagent, but they might not say where they got it from or what the catalog numbers are. So really we wanted a fully transparent, very detailed protocol where anyone who wants to do this assay can go find it on protocols.io and hopefully follow it step by step. And also sort of from a practical standpoint, it's a longer protocol. I mean, it has some, a few steps in it that need explaining. And so we thought that if we could publish it once and put it up in protocols.io, future papers down the road, when we use this assay, we don't have to explain the protocol over and over and take up a lot of space and say like materials and methods. Explaining it again and again on how we do it, we can reference the protocols.io paper or reference the PLOS ONE paper, and someone can go and find it very, very detailed exactly how we did it.
And if anything changes, in a couple of years, you can actually create a new version, right, of the protocol, and then cite just the latest version in which you tweak an amount or you change an equipment, right, or anything like that. So yeah, it can save you time, down the road.
Yeah.
Exactly. And so regarding the peer review, can you elaborate on how the feedback that you received from the peer reviewers at PLOS ONE help shaping the final version of the protocol, if at all?
Sure. So yeah, I guess we were fortunate in that the reviewers didn't have any technical aspects of the protocol that they needed to be changed or optimized. Really, a lot of what the peer review was just sort of cleaning up some concepts in the paper, which was very helpful. But one thing that I remember from the peer review process, which I thought was a very valuable addition, is one of the reviewers wanted an alignment made of all the different OspC sequences and where the different primers bind for the original PCR. but also for that one ASPE step that I mentioned earlier, where a specific primer finds the specific genotype, and they wanted to know where all these hybridization sites were. So I made an alignment of all the sequences and all the primers and where they hybridize. And actually, I thought that was a really good addition. We put it in as a supplemental because it's sort of a visual of how the entire assay works. And so I thought that was a really good idea and a good addition to the paper.
I would like us now to go and dig a little bit in the paper itself and the protocol itself. And I don't know if you can, tell the audience, what do you think are the most critical steps in performing the protocol?
Yeah, so like I said, it has those 5 steps. I think they're all probably, very, very critical. If you mess up one, it's going to have a knock-on effect for the other. But I would argue that maybe the most critical step is just that initial PCR, because that's the product that you're putting into the rest of the assay. So if you have a tick sample or any sort of sample, what you have to do first is amplify OspC. So it's a multiplex PCR. So any genotypes that are there are going to be amplified. And that part has to work, because if that part doesn't work, then the rest of the assay, you're not going to get an output. So you have to get a really robust initial PCR, confirm it with a gel, make sure you have a band, and then you can go with the rest of the process. But I would say you need that good starting point. Otherwise, the rest of the assay just isn't going to work for you.
And while developing the assay, was there anything that you tried and didn't work and didn't make it to the final version of the protocol?
Yeah, so I would say, the assay was sort of a culmination of a lot of optimization steps, and some helped. So those are the ones that made it in, and obviously some didn't help. So there were some things that we tried which just didn't improve the assay. And by improve, I typically mean increase the MFI signal. So that's the fluorescent intensity at the back end. We were trying to get a really good signal to noise ratio. And so, for example, one thing I can think of is we used all these primers, which were sort of adapted from that RLB assay. And I tried using different combinations of primers that target one strand of DNA, let's say the sense strand, compared to the other strand, the antisense strand. So trying to get longer extensions, which might equal higher signals. Because you do that incorporation of bioaccumulated nucleotides. So I was trying all sorts of different combinations, all one strand, all another strand, mixing and matching. And basically, anytime I went away from the original setup, which targets the sense strand, I'd get cross reactivity and weird false positives and things like that. I could just never make it better. So I spent a little bit of time trying this idea that I could mix and match primers. It didn't work. So we just went back to the original setup, which works great, gives you good values, and there's no cross reactivity.
And since you publish both the protocol and the article, have you come to a realization that there are steps that are could be or are a little bit finicky and might need a little bit of troubleshooting?
Potentially, I would say there's a lot of any sort of a lot of assays in microbiology and molecular biology labs. It can just be a little tedious. You have to be pretty good at pipetting, because it's a lot of transferring of small volumes of liquids to 96-well plates. A lot of transferring of liquids from plate to plate. So I would say one thing that can be a little tricky is you just have to sort of stay focused throughout the entire process. And one tip that I have is because you're going from multi-channel pipetter from plate to plate quite a few times, is I make little dash marks on the plate I'm going into, so I know what column I'm in. That way, if I sort of lose focus and I start daydreaming for a second, I can come back and figure out where I left off. So that's sort of just a little tip that people would learn after running it enough times. In terms of optimizing based on different labs, I'm not sure. I think you probably have to figure that out based on, the equipment, different thermal cyclers and whatever that you're using in your lab.
And before we wrap up, I think that my last question would be if you can tell us, as an early career researcher, what has been the significance of publishing this peer-reviewed paper for your career?
Yeah. So one thing that stands out to me is that this was my first authored manuscript. So it was the first time that I went through the whole process. So, I developed the assay with help from other people in the lab. And then I took the lead on writing the paper, incorporating all the other author comments and edits and revisions, and then submitting to PLOS ONE, and then addressing the reviewer comments. So I had been involved in that before as, like a middle author on a paper, but I had never been the first author. So it was the first time that I had done the whole process. So that was very illuminating for me and a good experience for me. And another thing is that, I spent some time on this during my PhD research, so it became a chapter in my dissertation. And so, of course, that helped me graduate as well. So yeah, this protocol has been, a milestone for me in my career.
And now, and before we really wrap up, is there anything else that you would like to discuss or mention that we haven't touched so far?
I don't really know. I mean, I'd say I hope other people use it. And if they make any tweaks or they improve upon it, I'd be curious to know what they do.
Patrick, again, thank you so much for your time, and I wish you success.
Thank you. Thank you for giving me the opportunity. This has been great.